So again, this is protein synthesis, and this is a continuation of your DNA replication, okay? We've talked about your DNA replication. In here, we said your DNA makes copies of itself. We said your DNA here, the N bases would follow the base pairing rule, okay? A would always be paired with T and vice versa. C would always be paired with G and vice versa, okay? This happens in the nucleus of the cell. You know that your DNA is found in the nucleus of the cell. Now, from your DNA molecule, the next step is going to be transcription. Okay, transcription here, one strand of DNA is used as a template in making RNA. Okay, so transcription here is a process through which RNAs are made, and we use one strand of DNA as a template. Okay, now the next process would be translation. Translation here, what happens in your translation is that information from your mRNA, from your RNA in transcription, it will be used to make proteins. Okay, we are going to the different steps of the different processes that we have here in the following slides. So again, how do we convert your DNA into the different genes that you have, into the different traits that you have? Whether you are going to be short, you're going to be tall, you're going to have thick lashes, thin lashes. How do we do this, or how do the cells do this? We call this process your protein synthesis. Okay, so this is protein synthesis, and this is a process that uses DNA to create an RNA, and RNA is going to be used in making of proteins. Okay, so protein synthesis actually has two parts. The first part is transcription. The second part will be translation. Okay, let me go to the next slide. Now here is the first part of your protein synthesis. Again, first part for this would be your transcription. What happens here is that first you have your RNA polymerase. It's like your DNA polymerase in your DNA replication. This is an enzyme that catalyzes the formation of RNA on the DNA template. It binds to the promoter, which is a specific nucleotide sequence of DNA, and starts transcription. Okay, so you have a DNA molecule here. Okay, so these two strands are the two strands of your DNA molecule. Now, what happens is that you have your RNA polymerase, a type of your enzyme. It is going to get attached to the promoter, one part of the nucleotide sequence of your DNA, and it is going to start transcription. Okay, it will start transcription. Now we go to the second step. The RNA polymerase adds RNA nucleotides that are complementary to one side of your DNA molecule. The RNA nucleotide follows base pair rules just as DNA, except that in RNA, there is no thymine. Okay, thymine is replaced by uracil. Okay, this is one very important difference between your DNA and your RNA. DNA, remember in your DNA, you have C and G or vice versa. You also have A and T, AT or vice versa. Now in your RNA, making of RNA, it would be AU. Okay, it will be AU, not AT. Because RNA has no thymine, it has uracil instead. Any questions? Okay, so again, the first step of your transcription, your RNA molecule is going to be formed by your RNA polymerase. As you can see here, only one strand of your DNA molecule is going to be used. Your blue colored structure here is your DNA. It has two strands. Your RNA, on the other hand, it only has one strand. So again, in your transcription, one strand, a single strand of your DNA molecule will be used as a template to make your RNA molecule. Now, as you can see here, if you have A in your DNA molecule, you cannot pair that up with a T. Because remember, this is an RNA. RNAs do not have T. Okay? RNAs do not have thymine. Instead of thymine, it has uracil. Okay, that is the main difference between your RNA and your DNA. Your RNA do not have thymine. They only have uracil. Okay, now we go to the last step. And so your RNA polymerase eventually would reach a termination signal or a stop and releases a DNA and your newly formed RNA molecule. Okay, so again, in your transcription, the making of your RNA using a strand of your DNA, remember that you also follow your base pairing rule in your N bases, but instead of AT, you have AU. Okay, you have AU instead of AT because your RNA do not have your thymine, they only have uracil. Any questions before I go to the next slide? Okay, now again, we're done with your DNA replication. DNA makes a copy of itself. It happens in your nucleus. Your transcription also happens in the nucleus. A single strand of DNA is going to be used to make a template. As a template to make your RNA, remember it's AU instead of AT, okay? Because RNA, again, has no thymine. Instead of thymine, it uses uracil, okay? Now, after transcription, what happens is that you'll have translation. If you already have your RNA that was actually made using your DNA as a template, okay, what happens is that the message in your RNA now is going to be used to make proteins, and you call that process translation. Okay, you'd call that process translation. Now, there are 3 different types of RNA before we get to your translation, the different steps of translation. You have 3 different types of RNA here. Your mRNA, that's the messenger RNA. It contains your genetic information in an RNA. So what was actually produced in transcription, the making of RNA, was your messenger RNA. Okay, you also have your tRNA or transfer RNA, which carries amino acids to the ribosomes to make proteins. I'm going to show you how it's done later. Now, of course, you also have the 3rd type, which is your rRNA or ribosomal RNA, which forms a part of your ribosome. Remember your ribosome, this is the organelle where the proteins are made. Okay, these are naked organelles where proteins are made, and your ribosomal RNA would form part of your ribosome. Okay, now again, going back to your chart that we have here, your illustration here, DNA replication, then transcription, then translation. Now, the different steps of translation here, again, translation is the making of proteins out of your RNA. There will be initiation, there's elongation, and there's termination. In initiation here, your mRNA binds with a ribosome unit. Okay, so your mRNA binds with your ribosome. Again, remember your ribosome is where your proteins are going to be produced. Okay, now the second process is elongation. Amino acids are brought to the ribosome by your tRNA. This is your tRNA or transfer RNA, and it's going to make or to transfer your amino acids. Then for the 3rd step, you have termination. The chains of amino acids are released, forming a polypeptide, a chain of your amino acids, okay? Then, of course, this amino acid sequence would form your polypeptide, and these polypeptide are going to be shaped into the different proteins. Remember, we have 4 different shapes of proteins, and the functions of your proteins would be dependent upon their shape. Okay, so you have your primary shape, which is just linear. You have your secondary shape, tertiary, and of course, you also have your quaternary shape of your proteins. Okay, so that's translation, making of proteins out of your messenger RNA. Now, how does it happen? Okay, we'd usually use in our study of translation in biology, we'd use your genetic code. Okay, this refers to how the nucleotide sequences correspond to a particular amino acid. Each set of 3 nucleotides would be called a codon. Okay, now your codon would always code for a particular amino acid, a stop signal or a start signal. Remember, you have 20 different types of amino acids. Amino acids are the building blocks of your proteins. Okay, so each of your codon, when you say codon, there are 3 nucleotide sequences. These are the 3 letters that you find in your messenger RNA. It might be AUA, AGU, it might be UGA, or whatever. Okay, we are going to take a look at the different examples of that later. And we use a codon chart to determine what specific amino acid or signal is expressed from a specific mRNA sequence. Now, this here is an example of your codon chart. Okay, so this is your codon chart. Pay attention. We work with your codon chart from the center going outwards. Okay, remember your codon would always have 3 letters. These are the 3 letters that you find in your RNA. That's why we don't find any T here. There's no T, there is no thymine, because of course your RNA has no thymine. Okay, we only have A, U, C, and G. Okay, remember your RNA has no thymine. Instead of thymine, it has uracil. Now again, I told you we work from the center going outwards. So if you are looking at your codon wheel here, Your GCA, for example, if those are the 3 letters of your codon, your GCA, that means that is going to code for your ALA amino acid. Okay, this is alanine, a type of amino acid. Again, remember we have 20 different types of amino acid. If say your codon has UCU, okay, if it has UCU, that means it is coding for SER, S-E-R, that's serine, one type of your amino acid. If your codon, for example, has CCC, okay, if it has CCC, that means it is coding for pro, another type of your amino acid. If it has ACU, for example, that means it is coding for THR, that's threonine, okay, another type of your amino acid. Okay, so again, you read from the center going outwards. Now say you have this as a different codons that we have. Take a look at your screen. Okay, you have this as a different codons that you have. Number 1 is AAA. If I will ask you what amino acid is being coded by AAA, what will be your answer? This, the what? LYS. Okay, very good. Okay, so Ivonte said LYS. The rest of you in your chat box are you're saying LYS. That's correct. Okay, very good. So that's AAA. That's LYS. That's lysine. Okay, one type of your amino acid. What about number 2, CUU? What is CUU? What is CUU? Okay, LEU, Zaria, that's correct. Donovan, Adriana, that's correct. Okay, so that's LEU. CUU, that's CUU. So that's LEU. Okay, that's leucine, another type of your amino acid. What about number 3, GAU? What's GAU? Okay, GAU, that's ASP, that's correct. Okay, so ASP, that's your aspartic acid, another type of your amino acid. Okay, so that's GAU. What about, so your number 2 again is leucine, number 3 is aspartic acid, that's ASP. What about number 4, UGG? What's UGG? Okay, Zaria said TRP. Adriana says the same thing. Donovan, the same thing. So again, number 4, that's UGG. So that's TRP. That's correct. That's your tryptophan. Okay, another type of your nucleic acid. Or not, not nucleic acid, amino acid, I mean. What about number 5, UGA? What's UGA? Okay, that's the stop codon. That's correct. So UGA, that would be your stop codon. That means whenever your tRNA sees the stop codon, it's going to stop coding for that protein sequence, okay? And then it's going to chop off that sequence of amino acid, that sequence of polypeptide, and it's going to form it into proteins, okay? So that is how you interpret your codon wheel. Now, sometimes it does not, you won't see it in this form, okay? We see a codon wheel here. Sometimes you see it in this form, okay? Sometimes it's in a chart. Okay, but you can still interpret this. You have your first letter here, second letter is right here, and then you have the third letter here. So say if I am looking at UCU, okay, if I'm looking at UCU, that's U for the first letter. So these are the different amino acids that I'm going to consider. Again, I said UCU, so that's U. Second letter is C, and first letter is U. Okay, so that means that would be serine. Okay, got it? So that's UCU. Now say I have CAG. If I am looking for CAG, that would be C, second letter is A. Okay, so A is right here, and G is right here. So that's glutamine. Okay, so CAG is glutamine. Okay, now what about if I'm looking for ACG? What will be ACG? ACG. What will be ACG, people? Uh-huh. Okay. A, C, G. So A is right here, first letter A. C is right here, and that's G. So that means that should be threonine. Okay, so Nasir and Donovan are correct. ACG, that's threonine. Okay, now what if you are looking for GAU? GAU. What's GAU? Okay, so Donovan says aspartate. Adriana says aspartate. Azaria, the same thing. Okay, so again, we are looking for GAU. So this is G, first letter G, second letter is A, and third letter is U. So that would be GAU, and that would be the correct thing. Okay, so that's correct. That should be GAU, aspartate. Okay, now what if I'm looking for AUG? AUG, what's the answer for AUG? What amino acid is AUG? AUG, thank you. Okay, methionine, that's correct. Nasir says methionine, Jason says the same thing, Adriana the same thing. So AUG, that's A, then U, then G, that's methionine, that's correct. Okay, now you see that there is a word start there. Your methionine, AUG, that's actually your start codon. Okay, so if you have a long chain of codons, you have a long chain of your nucleotides, the different letters in your RNA, and you are going to translate that, you are going to make that into your protein, you'd always look for your AUG. Okay, AUG would always be the start codon. You don't start with anything else but AUG. We are going to have some exercises on this tomorrow. Okay, not today, since we do not have enough time. So again, your start codon, that's AUG, and that is going to be your methionine. Now, what about your stop codon? For your stop codons, you actually have three types for this. Okay, so that's stop, this is also stop. Who can give us the different letters that can represent your stop codons? Okay, there should be three different stop codons with three different sets of letters. Who can give us the letters? This time you give me the letters, okay? Do not give me the name, give me the letters. I'm asking for the stop codons. Okay, Nasir says UAA, that's correct. What else? Okay, Jason, Adriana, Nasir gave all three. Okay, so UAA, UAG, UGA, and those are all correct. Okay, so again, for your start codon, there's only one start codon. You'd always look for your AUG. I'm going to give you an example on this tomorrow. I'm also going to ask you to do this tomorrow. Now, for your stop codons, you have three types. Your UAA, so U, A, and A. Okay, that's your stop codon, the first stop codon that you have. You also have UGA. Okay, that's another stop codon. And you also have UAG. Okay, so those are the three stop codons that you have. So again, whenever you have a long chain of your nucleotides, you'd always look for your AUG and you start coding there. Okay, you start translating, you start making your proteins with your AUG. That's your start codon. You'd always start with that. Okay, if you don't see your AUG, you don't start translation. Okay, translation again is using your information in your mRNA, your messenger RNA, to make proteins. Now, once you see any of these three, your AAA, UGA, or UAG, then you stop coding. Okay, we'll have some activities on this tomorrow. All right, so again, these are the different processes that we studied today. We started with your DNA replication. Again, remember, DNA replication is a semi-conservative process. From a single DNA molecule, you come up with two molecules of DNA. We call it semi-conservative because one strand came from the old DNA molecule. The other strand is entirely new. Remember that we follow the base pairing rule. Okay, so A would always be paired with T and vice versa. C would always be paired with G and vice versa. Okay, Chargaff's rule tell us that whatever the number of T that we have, that would also be the number of A's that we have. Whatever is the number of C that we have, that would also be equal to the number of G's that you have. Now, from your DNA replication, from your DNA, one strand of your DNA here is going to be used as a template to make your mRNA, and you call that process transcription. So in here, you can see you have your TAC, TAG, AGC, ATT as the different nucleotides of your DNA, and these are the complementary bases of your RNA. Notice again that instead of T, you use U. Okay, remember there is no thymine in RNA. We use uracil instead. Okay, so you have AU, but you still have TA. Okay, there's no problem with A. Your RNA still has an adenine, but there is no thymine, so we don't see any T's in your RNA. Okay, so these here are the complementary bases of your RNA. This now is the sequence of your nucleotides of your RNA. You got this from your template, which was one strand of your DNA. Notice the difference between your RNA and your DNA. DNA is made up of 2 strands. RNA only has 1 strand. DNA has thymine. Okay, RNA on the other hand has uracil. It has no thymine. Now, from your RNA, you are going to use your genetic code, the different codes that we have that we studied a while ago. You group your nucleotide bases here, or your nitrogen bases, by 3s. You call each of these your codon, and you'd always find your AUG. Remember, AUG is methionine. You'd always start with that. Okay, so you continue, you start with your methionine, then you have AUC, that would give you ILE, then you have UCG, that would give you SER. Now remember, your stop codons, it can either be UAA, it can be UAA, UGA, or UAG. Okay, any of those 3. As soon as you see any of those 3, you stop coding. That's it. So that means this is now the chain of your polypeptide that you've made out of your RNA. Okay, and this chain is going to become your proteins. And this chain, of course, that's going to dictate the different traits, the character traits that you have, that we see. Okay, whenever you look at yourself in the mirror, those are dictated by your DNA that was, of course, used to make your RNA, that was, of course, used to make your proteins.